describe blood vessels. Arteries carry blood that leaves the heart, while veins return blood. And there's a really good kind of tutorial or graphical, if you like watching videos to learn about anatomy, you go to watchlearnlive.heart.org. It's a really good tutorial on heart anatomy, heart valves, and kind of the things that we'll talk about today. So that we'll post a link to those resources as well. All right. So I think one thing I want you to notice is here, you usually think of arteries as carrying oxygenated blood, so you'd make them red, right, like the aorta. Here we have the pulmonary artery, and it's blue. And that's because it's sending blood away from the heart that's going to the lungs. So this blood doesn't have oxygen, but it's still an artery. So that's kind of the one exception where you don't see an artery that's colored red. It's colored blue because it doesn't have oxygenated blood. All right, so once blood comes into the right atrium, it sends blood to the right ventricle. And it's sending that blood through a one-way valve. So the blood can it's kind of like a one-way street, right? So blood can go one way, but it can't come back the other way into the right atrium. And if it does, then it's called regurgitation. That lowers heart pumping efficiency, right? Because when the right ventricle squeezes, it's not sending the blood to the lungs, it's sending some right back up into the atrium. So you're not getting as much blood flowing through the heart. And so that's depending on how bad it is, it can be something that will need surgery or a valve replacement. And so why does the valve open? It's kind of a passive thing. It's not like there's muscles that are yanking the valve open. You do see papillary muscles right here, but it's really that the pressure in the right atrium is increasing enough to open the tricuspid valve, and that's allowing the blood to enter into the right ventricle. What those papillary muscles are doing are keeping the valve from opening back up the other way. So that's called prolapse. So if the valve goes the wrong way, you got to get an inversion. Then again, it's a very serious condition where your pump's not going to be able to, the heart's not going to be able to pump efficiently. You're going to get blood going the wrong way. So you shouldn't think about the papillary muscles as opening and closing the valve. They're just there to kind of stabilize it and keep the blood from going backwards. All right, so once the tricuspid valve opens, and you should know this, so this is great material for a test, right? It's super easy to ask about, and we covered it. So I'll also try as we go along to kind of identify things you should be paying attention to. Do you have a question? Oh yeah, that's how you. All right. Feel free to interrupt if you do have questions. Okay, so once the tricuspid valve opens, blood fills the right ventricle until the opening of the pulmonary valve. So the pulmonary valve is going to be the valve that opens and allows blood to leave the right ventricle and go to the lungs, where it's going to collect oxygen. The pulmonary valve has 3 leaflets, and those open when the pressure in the right ventricle increases to a certain level. Now, if we look here, the pulmonary artery has 2 branches, right? One for each lung. And then there's this little one here that connects to the aorta. And that's called the ductus, or the ductus arteriosus. So in fetal development, the fetus has lungs that are full of fluid. And so there's no reason for the blood to be going through the lungs and collecting no oxygen, right? The fetus is getting oxygen from the mother. So instead, it shunts. And so there's a path for the blood to go straight from the right ventricle and back into the aorta. So it has its own circulation, it's just not going through its own lungs. It's getting its blood through the umbilical cord. And there's also a second shunt that's through the 2 atria. So the blood can go straight through. Only about 10% of a fetus's blood goes through its own lungs. Then when the baby's born, then that closes. And so the pressure, when that baby takes its first breath, so I don't know if you've ever, probably not, seen a baby be born, but they're really concerned with whether or not that baby takes its first breath. And that first breath is really important for decreasing the pressure around the pulmonary artery, and that'll close the ductus arteriosus, and that'll turn into the ligamentum arteriosum. And so it's going to close, and you're no longer going to have a shunt between the pulmonary artery and the aorta. If it doesn't close, then this is one of the ways that the hospital makes a lot of money, is that the baby's going to have to go to the NICU, and they're going to have to close that back up. So it's called a patent ductus arteriosus, so a PDA. And so that's one of the main things that they have to treat newborns for, is so they still have that shunt, and that shunt's preventing the heart from working efficiently and cycling blood to the lungs and then back out to the body. So it's this kind of crazy sequence of events that happens right as soon as the baby's born that closes off their fetal circulation and opens up their normal circulation. All right, so once the blood gets oxygen from the lungs, then it comes back through the pulmonary veins. So again here, this is a situation where you have a red-colored vein, right? So usually your veins are blue, right? You look at your arm, you see blue veins. Here you have red veins, right? Because the veins are coming back in. So these are the two examples where they're opposite. It enters the left atrium, and it goes through the mitral valve. Now, why do you think that the mitral valve looks different? First off, what's different about the mitral valve than the previous valves we've looked at?
Bicuspid.
Right, it's bicuspid. Why do you think it would have to be bicuspid instead of tricuspid? Yep. Right. Yeah, so there's going to be a lot more pressure in the left ventricle, right? So right ventricle's got to send blood to the lungs, and it comes back. It's a short little trip, right? Heart's here, lungs here. It is not a ton of work. Toes are quite a bit further away, right? So you got to put a lot of pressure from the left ventricle, get blood all the way to your brain, to your toes, all the different parts of you that you want to be have circulation. So you're going to have a lot more pressure. With 2 leaflets, it's going to be a lot harder to open them up. With 3 leaflets, it's going to be a lot easier. And so kind of the prevailing theory is that you need, because you have higher pressures, you only want 2 leaflets in your mitral valve. Finally, blood enters the left ventricle, and it stays there until the aortic valve's open, and then blood goes to the aorta and out to the rest of the circulation. And so here's a picture of the aortic valve. So heart sounds. So if you've ever put on the stethoscope, you've heard the lub-dub. Those are called S1 and S2. So let's see if I can play. Maybe this works. Turn this up, and then let me make sure my computer's not muted. Oh, it is muted. Is that coming through the, I don't know, is that just my computer, or is it coming through the, you don't hear it, do you?
I think it's just quiet, but I think it's through the projector.
Okay, no, let me turn it up. I'm just trying, you can kind of hear it, right? Okay, let's see if it works. In Broward 12 it would have worked, but I don't think we got that one. That's too bad. I think it's letting me play it. Okay, so I'll put this on. So basically you want to be listening to, what I'll do is I'll post this so that you can listen to it. But basically you want to hear these 2 sounds. And so the first one is going to be, the heart sounds are basically the closing of the heart valves. So when they slam shut, you can hear that on the stethoscope. the first sound is the closing of the mitral and tricuspid valve. And so most of that sound of S1 is due to the mitral valve. And that's because it's closing in response to the most pressure. So it's slamming the hardest. They're slamming right about the same time. And so most of the sound you're going to get is from that closing. The second sound is the closing of the atrial and pulmonic valve, or the pulmonary valve. And so you get the two chambers, right? So you got the atria and the ventricles, and they're both left and right are contracting at the same time, or approximately the same time, and so you're hearing those two sounds together. And if you put the stethoscope at different parts, if you've ever gone to, like for your physical, when you do sports, they put the stethoscope in different places, right? So they don't just leave it in one place. And so there's 4 different places on the torso that you can put a stethoscope. And so the two first ones are up toward the top. And so the base right, that's this one. And so if you haven't looked at these diagrams, you always look from the patient's point of view, right? So this is base right, but it's on the left. That's because if the patient was lying down, it would be the patient's right. So every time we say right atrium, we're putting it on the left. That's because you're always looking at it from the patient's perspective. So base one is gonna, or base right, is gonna reflect sound from the aortic valve. Base left is gonna give you sound from the pulmonary valve. The lower left sternal border is gonna let you listen to a tricuspid valve. And the fourth is gonna mostly give you sound from the mitral valve. The mitral valve is so strong that you can only really hear the tricuspid valve when you go to left sternal. So if you put your stethoscope down here, you're not really going to be able to hear any sound coming from the tricuspid valve. So that's one way to separate it. One way that a doctor knows that something's wrong is sometimes they hear a whoosh, right? So that can be a sign of atrial septal defect. So the wall between the two atria maybe isn't totally formed, and they can hear the blood going through there. Another thing they listen for are splitting of sounds. And so when you breathe, the pressure in your lungs is going to change the pressure in your pulmonary veins and pulmonary arteries. That's going to change the timing of when the pulmonary valve is going to close. And so what they're listening for is if that sound stays separated. So if you're breathing and then sometimes it separates when you breathe in and it comes back together when you breathe out, that's physiological. So that's called a split S2, and it's coming apart and coming together along with the breathing. If it stays separated, then that's telling them that there's some dysynchrony between the different chambers of the heart, and that there's something that they have to look into. And that can be range from a lot of different things, everything from conduction problems, so the electrophysiology of the heart might have a problem, all the way to heart failure and pulmonary hypertension. So you can imagine how a split S2, which is reflecting the pressure in the pulmonary valves, is going to maybe be affected if you have high blood pressure in the pulmonary circulation. All right, another important part, so we've talked about kind of the big parts, right? So the vena cava are going to collect the blood from the body, send it to the right ventricle, it's going to go to the lungs, come back, and then to the rest of the body. But we also need to supply the heart with its own blood supply. And these are going to be the coronary arteries. So coronary arteries are going to branch off early on from the aorta. You can imagine that there's going to be a problem if those get blocked off, right? So we've talked about how important it is for the blood, for the heart to keep functioning. If the heart doesn't have blood supply, then certain parts of the heart are going to die. And so that's really what a heart attack is an occlusion of the coronary artery. And so the left anterior descending artery is a commonly used model for a heart attack. And so in animal models, what they'll do is they'll suture off the LAD and they'll cause an infarction. So some cells to die in this area. That's a way that they study heart attacks and also arrhythmias. And so the posterior descending artery, which is over here, will supply the right ventricle. The left anterior descending artery is going to supply the left ventricle. Does that make sense? Okay. And so we talked about coronary artery disease. And I think we said that's 370,000 people a year die from coronary artery disease. And you can see it. So this is a guide wire that they put up into a person's heart. And what they'll do is they'll try to inflate a balloon to unclog this artery. So they put some contrast in there. And you can see that it filled all the different veins in the heart quite well. This is with a fluoroscopy that they're seeing this. This is the wire that they used to put in the contrast and guide it there. You can see here, right here, you have kind of a faded spot. And that's an occlusion in the artery that's preventing the rest of the heart from getting the oxygenated blood that it needs. And so what they'll do is an angioplasty. And you've probably heard about this, where they'll go in and they'll try and open those up. And so the most straightforward thing they can do is they can put in this wire, inflate a balloon, crush the plaque, and open up the artery. The problem is that within months or a couple years, it'll close right back up. And the person will tend to have the exact same problem. So they haven't really cured the disease, they've just temporarily opened it up. And so one thing that's gained a lot of attention in recent years is using stents. And so the first generation of stents was a device kind of like this, where it's a wire mesh, where they just put it in there, they open it up, and then the idea is that it'll keep the artery open. And what they noticed was that the plaque would reform around the mesh and close the artery right back up. So even though they were holding it open, the plaque would grow right around it. And so the most recent iteration of stents is using a drug-eluting stent. So they add drug, they have drugs included with the stent that are supposed to prevent the artery from narrowing again. Yep.
With the balloon, is it pushing the plaque further downstream, or is it just smashing the plaque against the blood vessel?
The idea that it smashes it and then it goes downstream, but it's not like it's trapping it there permanently. And then they'll deflate it, right? Because you can't just leave the balloon in there. They would leave the stent in. But even with these drug-eluting stents, there's been some really big papers, probably in the last year or two, that have shown that they're not very effective. And so this is kind of an active area of biomedical development. I think if anybody came up with a stent that was effective and you could leave that in there for 10 years, it would radically transform cardiac practice.
By not very effective, you mean the plaque just forms again?
Yeah, so there's a lot of enthusiasm. They thought, okay, we're going to add drugs to the stent, keep the plaque from reforming. Some of the initial studies were very promising, but the most recent studies showed that they weren't really very effective at all, which is pretty disappointing and also hits the pocketbook of a lot of cardiologists.
What was the lifetime on them with being ineffective?
I think it's a couple years. I can look that up. I just thought of that paper now, but I can. What I'll do is I'll post that paper on as a resource if people want to look at that. All right. So, how much time do we have? Oh, we have a good amount of time. All right. So let's talk about heart control systems. So what's going to control that? I know that the plumbing is kind of painful. It's not as bad as lungs, but it's a little bit painful. So let's talk about what controls the heartbeat. So probably the most important regulator of the heartbeat is the autonomic nervous system. So you can decide to stop breathing, right? At least until you can't take it, right? You just stop breathing. You don't say, okay, I'm going to start my heartbeat for a little bit, right? It keeps going. It's not something that you have direct control of with your conscious mind. And that's because it kind of oscillates on its own. It beats. And so what your body does control is the rate. It says beat faster or beat slower, but it never says stop beating. So that would be deadly. So you have an oscillating heart. Sometimes you want it to beat faster. Sometimes you want it to beat slower. The main way that the body controls this is through the autonomic nervous system. And so there's two branches of the autonomic nervous system. One is sympathetic. The sympathetic nervous system coordinates the response when you need a lot of energy. So you're running a race, you're running late to class, you're taking an exam, you need to run away from a tiger, right? So that's the main, if you think back from an evolutionary point of view, you want to be able to flee quickly, you want to be able to respond quickly to survive. And so these are pretty primitive systems. The parasympathetic system is more at rest, right? So you don't want to use a lot of energy, you want to digest food, you want to eat, you want to sleep, and so you want to use less energy. And so they're kind of in opposition to each other all the time. And you can kind of see, if you see the response to the different organs, so when you have a big sympathetic response, this is kind of fight or flight response, you have dilated pupils, less salivation, right? You're not thinking about eating, so there's no reason to have a lot of salivation. You get bronchial dilation, right? So your lungs are primed to take in more oxygen. You're not thinking about digesting food, so it shuts down your stomach. So I don't know, if you go for a long run, you're not really hungry, right? Like you can be hungry when you leave, but when you come back, it's about the last thing you want to do. Your liver holds a huge amount of glucose. It's going to start releasing glycogen to supply all the muscles. You're going to get epinephrine and norepinephrine release. That's going to affect your heart rate. It's going to relax the bladder, so you're not going to want to go pee. We had a little dog, my wife did when I met her. And if you looked at the dog, he wouldn't go. Like, he just couldn't. He was super shy. He'd get really nervous. And it wasn't his fault. It was a sympathetic reaction. He just got nervous and everything closed down. And so that's what's going on. This parasympathetic, this is when you're relaxing, right? So you're going to constrict the pupils. You don't need to see as acutely or get as much light in. You're going to decrease the heart rate. You get bronchial constriction. You're going to stimulate digestion, right? So now your body's not worried about running away from a tiger. It's thinking about eating. You're going to relax. You're going to sleep. You're going to be able to go to the bathroom. And then there's also, kind of for sexual stimulation, you have a parasympathetic response first, followed by a sympathetic surge. And so that it's important to be able to relax first and then kind of get toward it. Use that to great effect. So for the heart, we're going to talk about exactly what controls its beating harder and beating faster, right? So for heart rate, you're going to want to be able to send more blood to the rest of the body, so you want to beat faster. And it's not just faster, you also want to beat harder, so more blood per beat. And so it's kind of interesting because the innervation is different. So parasympathetic will control kind of the pacemaker, or the part of the heart that's controlling its beating rate, while the sympathetic innervation is going to be all throughout. Yep?
Like medications that will increase your heart rate, will they automatically activate the sympathetic reactions that you're talking about?
A lot of them will. So yeah, so that's a consequence, right? So if you're stimulating the sympathetic nervous system, cocaine, then that you'll get an increased heart rate. But there's also direct ways that you can affect heart rate as well. So some antiarrhythmics will actually act directly on the cells that control the heart rate and slow that down. And so there's multiple ways to do it, but that's one way.
Like steroids, will they also, they'll increase your heart rate, but will they, all the other things that you mentioned?
Well, so steroids are pretty potent signaling molecules. So my guess, and I don't know this for sure, I can look at it, is that they're acting directly through the sympathetic nervous system. So those are hormones. So basically, hormones are neurotransmitters that go throughout your whole body, right? So neurotransmitters are for local control. Hormones are for controlling your whole body's response. So yeah, I would think that they'd be interacting with that system.
So you don't need the vagus nerve to, the pacemaker cells can operate independently of the vagus cells. They just modulate the rate of...
Absolutely.
Right.
Yeah, so we'll get to that in a couple slides. But yeah, it's a great question. So let's talk a little bit about kind of the heart's control system. So you've seen the ECG, right? So you put a couple of electrodes on the chest, and you see these blips. They look something like this guy on the bottom, right? And so you see that on TV. And that's because your heart's producing electricity, just like your brain, which is reflected in the EEG. Your heart's also producing electricity, and that's what's telling it to contract. It's also synchronizing the contraction of the different chambers. So up in the right atrium, you have the SA node. And what the SA node is a group of cells that are oscillating, right? So their membrane potential is changing up and down, up and down. And their role isn't to contract. They're not controlling any of the heart's beating. They're just creating this oscillatory wave. And that's spreading through the heart and telling the different chambers to contract. So the kind of the control system of the heart is right here in the right atrium. And so if we look at kind of the cells that control the heart, you have the SA node, and that's what's oscillating initially. And then it's communicating with the AV node. The AV node conduction is very slow, right? So first what you're going to do is you're going to contract the atria, and then you want to wait for the ventricles to contract. Do you know why you want to wait?
The blood to fill into the ventricle.
Right. Yeah. You need to give it time to fill up. And so there's going to be a delay here in the AV node. So the atria and the ventricles aren't electrically connected except through the AV node. If they are electrically connected, then that's a syndrome called Wolff-Parkinson-White. And so what a doctor will do is put a catheter in there and it'll burn the part of the tissue that's connecting the two chambers. So it's actually a pathology if they get connected in any way except through the AV node. The Bachmann bundle is a system of fibers that's going to innervate or go through the left atrium. And so the signal's going to spread pretty quickly over here and down through here, and it's going to synchronize the atrial contraction. Then it's going to slowly go through here, and then once it finally gets through the AV node, it's going to go through the His bundle, and it's going to simultaneously activate the contraction of the right and the left ventricle. And so that's going to go through the Purkinje fibers. The autonomic control is focused on the pacemaker, right? So in particular, we're looking at, it's not like it's making the ventricles beat faster directly. It's making sympathetic stimulation is going to make it beat harder, but that sympathetic stimulation that's directly interacting with ventricular tissue isn't what's making it beat faster. What's making the heart beat faster is its control over the pacemaker potential. So if you look at it here, this is the kind of the dotted line in black is the normal beating rate. That's kind of what the action potentials would look like. So if you measured the potential in the cell relative to the ground and looked at the pattern, you just see that it goes up and down at a certain rate. So this is about 60 beats per minute. When you apply acetylcholine, which is a vagal stimulus, you'll see that the rate of rise of that potential is going to slow, and that's going to slow down the beating rate of the SA node. Conversely, epinephrine will speed up this rate of rise and it'll start oscillating a lot more quickly. So one thing we'll talk about in a future lecture is exactly how this works. So what proteins are controlling that beating rate and how is the autonomic nervous system affecting that as well. Does that answer your question? All right, so what does the SA node look like? It actually has a lot of structure to it. So people used to think that it looked like this. This is the textbook SA node. You just got a little ball of cells there. Instead, through computer modeling, people have shown that it has a ton of structure where it has kind of these cells around it that block conduction. And so, have any of you ever tried to start like the wave at a baseball stadium? So, I mean, the first thing that happens, right, you get someone up there and they're going like this all by themselves, right, and it doesn't work, right? The whole stadium's not going to go just because one person's going like this. So first, they recruit like 10 of their friends, and they're like, okay, we're all going to do it. And if 10 people do it, then maybe you can get your one section that you're in to help, right? And they're going to start, and it's going to go a little bit, and it's going to stop. And eventually, you got to recruit a whole group of people, maybe all the way up and down, if it's going to propagate all the way around the stadium and keep going. And that's the same thing that's happening here. Is if you only have one cell, or a small group of cells that are oscillating and they don't recruit other cells, it's gonna be too much of an electrical mismatch, right? So you're gonna get some electrical excitation, all of it's gonna spread out to the other cells, and you're not gonna initiate a heartbeat. So what happens is it starts down here, and then it recruits a bunch of pacemaker cells until you have a critical mass, and then it spreads through the right atrium. And so this structure that allows the wavefront to build, just like when you do the wave at a stadium, is kind of critical for a small group of cells that are very tightly innervated and controlled by the autonomic nervous system to regulate the heartbeat. And then the other thing that's kind of, and this is kind of a newer concept, is that you don't necessarily have one area where your pacemaker always is. And so for a long time people thought you have your pacemaker and it's oscillating, and you have all these inputs and it's integrating those to control the heartbeat. What people have found is when they put different conditions, so let's say you have low sodium, then the pacemaker's over here. If you have low calcium, it's over there. And if you have adrenergic stimulation, it shifts down here. That's kind of interesting, because what that means is if you have different signaling components, different cells are taking over. So it's like you have specialized cells, and if you have beta adrenergic stimulation, then one group of cells is saying, okay, this is my specialty, I'm going to take over. I'm the group of cells that controls the heart when we have a fight or flight response. So this isn't totally defined as to how exactly this works, but it's kind of an intriguing idea, is that you don't just have one group of cells, you have several different specialized groups, and under different conditions, they take over in a beneficial way. Okay, so after the SA node gets excited, you get propagation through the atria. You get the pause in the AV node, and then you get this spread through the ventricles, which is quite rapid. And so that's kind of what you see here, right? So you get this green excitation here on the ECG that kind of corresponds to this first blip. That's called the P wave. Once the signal gets through the AV node and spreads through the ventricle, then you get this very strong depolarization, which is the excitation of the ventricle. So why is this so much bigger than this one? So why is the QRS, which is the ventricles getting depolarized, bigger than the P wave of the atria?
Ventricles are just bigger?
Yeah. Yeah, absolutely. So what the EKG is sensing is changes in potential, right? And so a much larger mass of tissue is going to be able to produce a much larger electrical signal relative to the much smaller atria. And you tend to have longer excitation, right? So you need to bring in more calcium. Calcium is what tells the heart cells to contract. And so you need more calcium, and you need a much stronger heartbeat. And so this is kind of an early EKG. Right now you wouldn't be able to do that. So these are totally uninsulated wires. So they would run the wires into the next room. If you did that now, you'd just see 60 hertz noise, right? Because we have light bulbs and cell phones and cell towers everywhere. So we have a lot of RF noise. But back then they could just run a bare wire back to their potentiometer. They can measure the EKG. So you just put your arms in salt water, and they could see the heartbeat. This is one of the first examples of someone getting their EKG. And this is the lead one. So this is exactly what's reflected here. So that's one of the labs that we'll do, I think it's the fourth lab, where you guys will do the vectrocardiogram, and you'll kind of learn how the EKG works. All right, so how does the electrical system or signal get from cell to cell, right? So you have these pacemakers, right, and they have to recruit the other cells until get them to contract as well. So you learned in the first semester about neurotransmitters, right? So you get neurotransmitter release. Did you learn this in any detail? Maybe? No? You did or didn't learn about neurotransmitters? Yeah? Okay. So it's a little bit different in muscle cells, where they're electrically connected. So there's gap junctions. So there's proteins that connect one cell to another, and if the potential in one cell is higher than the one downstream, then charged ions can flow across those connections, and they can change the potential in the adjacent cell. So you have the pacemaker cells get excited, and then that propagates to the cell next door, that excites that cell, and so on and so forth. And the propagation can spread throughout the heart. And you can see here, someone labeled them fluorescently, and you can see that they're kind of at the ends of the cell. So what do you think that means in terms of the cardiac conduction? You have fibers, right? So here's the fibers, they're going this way. And so what do you think the distribution means?
Single line, not like a, it's just it's like a 2D line of muscle cells.
So it's going to be directed, right? So if these are where your connections are, then the wave's going to travel in that direction. You're not going to get transverse conduction very much. Most of the conduction is going to go right down the fiber. So it's like the cells are stacked end to end, and the propagation is going to run down the fibers. You're not going to get uniform conduction down one way. And that's really allows the heart to kind of orient the fibers. So if you looked at the fiber orientation, they kind of orient in a turning pattern all the way across the heart wall. And that allows it to kind of counteract the pressure forces a little bit better. So because the pressure it's sensing on the inside is, or the pressure that needs to deal with on the inside of the heart is different from the outside of the heart, the fibers change directions on the way in. And so having the gap junctions on either side allow it to control the timing of the contraction even within the heart wall. And so connexins, if you look at them at a very microscopic level, you can see that they're formed by 12 different subunits. So it's like you have these 6 different subunits around there, and they kind of form a whole here. And then you have one group of 6 on one cell, you have another group of 6 on another cell. There's been a lot of people who spend a lot of time studying why connexins open and close. And what the conclusion is it really doesn't matter that they open and close, because they open and close at such range that's way outside the physiological parameters. So they look at, so connexins can close in response to pH. So let's say heart's ischemic, then they tend to close. By that time, the heart tissue is pretty much dead. It can close at very negative potentials, but those potentials never get reached. So the way you can think of them is they're mostly open all the time. And so far, nobody's found a good reason why they close. And that kind of makes sense for the heart, right? You don't really want them to close and stop the heartbeat. So you need a pretty extreme situation for them to close up. And then there's different kinds of connexins. So, and they're all kind of specialized. So connexin 45s, which is one kind of connexin, are found in the SA node, whereas connexin 43 are in the ventricle. And so connexin 45 tend to be they're smaller, but they have a smaller pore, so they pass less current, so that's usually for slower conduction, right? So in the AV node, connexin 45, right? So we talked about how conduction through the AV needs to be really slow to allow the ventricles to fill. You're going to get connexin 45, or the distribution of the connections between the cells is going to regulate whether you have fast conduction in the atria or the ventricles versus the AV node, where it's supposed to be really slow and comes through there. I'm going to skip this, and then this will be our last slide for the day. So we talked about
Well, there's some connections slower than others. Like, what about them makes the signal travel slower?
It's just how much current they can conduct. So they have a different structured pore, right? So you might have, the simplest way to think of it, one's bigger, right? So you can have more throughput through it. Whereas another one's maybe more narrow or maybe more selective to certain ions. And so only certain particles or ions can get through those. But yeah, it's been pretty puzzling to people why exactly there's different connexins, and there's still a lot of debate in the field. So in some sense that's frustrating, but they can also get a lot of funding because they say, we don't understand it, so we need millions of dollars to try and figure it out. So I think the thing I want you to leave you with today is the idea of myocardial infarction and arrhythmia. So we talked about heart attacks, right? So heart attack is when the coronary arteries get blocked, and then a region of the heart dies, or it becomes damaged. What happens then is you have an area where the heart wave travels differently. And this is kind of something that we're going to think about in this section, is what changes happen that allow the substrate to be pro-arrhythmic, right? We talked about this. It's like 3 billion times your heart doesn't have a problem. You have a heart attack, the part of the heart dies, and now you disrupt conduction. So instead of this uniform excitation of the heart, you have these areas of slower conduction. It starts to come back around. So you get this part excited, this part takes a while, and then it comes up, and finally it gets excited. By the time it comes out, the rest of the heart's relaxed. And it can take over, and you get this reentrant arrhythmia, where the heart kind of starts beating out of control. And most commonly, or almost always, with heart disease, the final common pathway is arrhythmia. So the heart's maybe not beating well, it's maybe not supplying blood as efficiently as it could. But the way that people finally pass away is usually in arrhythmia. So the electrical synchronization of the heart becomes disorganized. And that, in a large part, this kind of disorganized behavior is from the decoupling of the myocytes and discontinuity. So instead of having a smooth surface or a smooth tissue to propagate, now suddenly it's disordered and it's not as organized. And that's what finally leads to arrhythmia. So to keep with doing 20 minutes of office hours, I think we're going to finish up there. And then next lecture, what we'll talk about is models. So we'll talk about Hodgkin. So if you have notes about Hodgkin-Huxley, I'd review those prior to next lecture. We'll talk about how you use those models to use them from our heart.